Good morning, everyone. Now we are going to start the third plenary session, and it's my privilege to introduce the chairperson, Professor Yogen Singh. He is associated with the University of Delhi. His area of research interest is bacterial pathogenesis, bacterial protein toxins, signal transduction in bacteria, bacterial virulence factor, cell biology, host pathogen interaction. He did his PhD from Patel Chest Institute, University of Delhi. He served as scientist and chief scientist in CSIR IGIB, and he has received several prestigious awards like Best Mentor Award of Indian Institute of Science. He is JC Bose National Fellow, and he is fellow of different national and international societies like Indian National Science Academy, Association of Microbiologicals of India, National Academy of Science, National Academy of Science 2010. Now I hand over this mic to Professor Singh.
Thank you very much. So good morning, everybody. Plenary session 3, and the first talk is online. So let's see whether he is online. All right. So the first talk is by Professor Gautam Sethi. Can you bring him on the screen? Dr. Gautam, can you hear me?
Yes, I can hear you. Good morning.
Hello.
Hello. Yes. Good morning. I can hear you. Hello.
Professor Gautam Sethi, can you bring him on the screen? Dr. Gautam, can you hear me?
Yes, I can hear you. Good morning.
Hello. Good morning.
Hello. Yes. Good morning. I can hear you.
Welcome to this session. So,
Yes,
Dr. Gautam, talking on
Gautam
Sethi, targeting oncogenic transcription factor strategy for cancer therapy. So, Dr. Gautam, please start. Dr. Gautam, can you hear me?
Yes, I can hear you. Good morning.
Hello. Good morning.
Hello. Yes. Good morning. I can hear
Welcome to
you.
this session. So,
Yes,
Dr.
thank-
Gautam, talking on targeting oncogenic factor strategy for cancer therapy. Dr. Gautam, can you hear me?
Good morning. I can hear
To
you.
this session. So, Dr. Gautam, talking on
Good afternoon. Good morning. Can you see my screen? Good morning. Can you see my screen? Good morning. Can you see my screen? Good morning. Can you see my screen?
Can you speak a little loudly? We cannot hear you.
Okay. Good morning. Now can you hear?
Yes, we can. Thank you.
Okay. Okay. Thank you. Good morning to all of you. First of all, I will like to thank the organizing committee, Professor Alok, for his kind invitation in this prestigious meeting. And it's due to some urgent work, I could not attend the physical meeting, but today I will be presenting my talk online on targeting oncogenic transcription factor STAT3 for cancer therapy. So I will begin with a slide showing a wide global variation in cancer incidence and mortality. So if you see in this slide, on the left-hand side, we have US, and there are more cases of lung, breast, and prostate cancer in North America, which has an aging population. If we move to Europe, the trend remains more or less the same. And if we talk about Southeast Asia, including India, so we find that lung and oral cavity cancers are the most common among men, while cervical and breast cancer are the most common among women. So there was a study published in Science a few years back, which demonstrated that in addition to the various risk factors for cancer that we already know, which include environmental factors, genetic factors, and lifestyle-related factors, bad luck has been contributed to be an important risk factor or connection to cancer. And bad luck implies just a random mutations that occur during the process of DNA replication in non-cancerous stem cells. So this is the latest study published in Lancet in 2022, in which the authors have estimated the cancer burden that can be attributed to different behavioral, environmental, occupational, and metabolic risk factors. And what they found that the leading risk factors that can be attributed to cancer mortality worldwide were smoking, followed by alcohol use and high basal metabolic index. So based upon this study, it was again re-established that the leading risk factors contributing to global cancer burden in 2019 were behavioral, whereas metabolic risk factors saw the largest increase between 2010 and 2019. So this has been our working hypothesis from the last few years after I completed my postdoctoral training at MD Anderson Cancer Center under Professor Agarwal. So we have looked at the contribution of different lifestyle, environmental factors, and we expect that they play a very important role in the process of inflammation, and they can function by inducing acute inflammation, what is we call it as therapeutic inflammation, and chronic inflammation, which has been linked to various hallmarks of cancer, including tumor cell survival, proliferation, invasion, angiogenesis, etc. And this is what we call it as pathological inflammation. So in recent years, in my lab at NUS, we have looked at also the role of non-coding RNAs and how they can mediate the process of inflammation in various chronic disease. So this is our recent article showing the role of non-coding RNAs in regulation of inflammation and also the role of non-coding RNA and macrophage interaction in tumor progression. So recently, we have done a lot of work using prostate cancer as a model, which is the second most common cancer diagnosed among men worldwide. And it is the fifth leading cause of mortality. And if we talk in Singapore context, it is the third most common cancer affecting Singapore men and sixth leading cause of mortality. And in recent years, it has been found that prostate cancer along with breast cancer cases have risen sharply in Singapore. And for example, prostate cancer, it is now among the top 3 cancer, whereas previously it was at the 7th place almost 40 years ago. So what is a prostate cancer? So it arises in the prostate gland of man, and it's a slow growing tumor. And one of the most important risk factors is that of old age. And globally, I have shown you it is the second most common cancer and third most common cancer among Singapore men. And the screening involves combination of prostate-specific antigen levels and Gleason score. So if you look at the stages of human prostate cancer progression, it involves loss of basal lamina, where we have this inflammatory condition called prostate intraepithelial neoplasia, loss of basal lamina, and then we have this condition called invasive carcinoma, which is managed by treatment with anti-androgens. But unfortunately, this anti-androgen therapy is not curative, and prostate cancer acquires a state, what we call it as androgen independent state, and then undergoes metastasis. So if we look at the current risk factors for prostate cancer, it includes aging. So most of the patients are above 70 years of age. Family history, if you have a first-line relative, your risk may increase by twice, and ethnic origin. So initially, it was found to be more common among Western men, but of late, the incidence have been increasing in Southeast Asia as well. So if you look at the current therapies for prostate cancer, they are based on PSA level greater than 20, Gleason score more than 10, or metastatic disease. And if the Gleason score is less than 6, we put the patient on active surveillance. And then other options include surgery, radiation therapy, androgen deprivation therapy. But once the patients are non-responsive and we have this castration-resistant prostate cancer, there are very limited treatment options available, one being docetaxel, which is a microtubule inhibitor. So if you look at the current treatment therapies, they depend upon the clinical behavior of prostate tumor. It is organ confined, and we have radiation therapy, surgery. For locally advanced tumor, we have androgen deprivation therapy. But in case of CRPC, there are not many chemotherapy and targeted therapies available, and hence there is an urgent need for identification of pharmacological agents to improve the existing treatment modalities. And agents derived from Mother Nature can serve as important source of anti-cancer drug, and that is also one of the main theme of this conference. So recently we have edited a number of special issues in prestigious journals like seminars in cancer biology, looking at the molecular targets of natural compounds for cancer treatment and prevention. And this is one of the latest one, which we have edited in May earlier this year. So today I will share the work done by my lab on this plant, which is indigenous to the Asian subcontinent. It is known as neem, or Azadirachta indica, and it is also known as Wonder Tree, Nature's drugstore or village pharmacy. And we already have a number of neem products in the market which have been used for management of different conditions, and also a wide variety of bioactives have been identified from neem, which include nimbolide, nimbane, etc., which have been used for management of different diseases, including respiratory diseases, inflammatory conditions, etc. So we have compiled a number of articles looking at the anti-cancer potential of neem for the management of different diseases, and also strategies to evaluate the pharmacokinetic potential and how we can improve the pharmacokinetic potential of nimbolide to accelerate its clinical development. So there are several reports that have looked into the anti-cancer effects of nimbolide in different cancers, including prostate cancer, where it has been shown to induce apoptosis. But in our 2 studies that we have published, we have looked at the anti-cancer effects in detail using prostate cancer model, cell lines, and transgenic as well as orthotopic model. So I will quickly go through these studies. So first I will start with the in vitro studies. So first we look at the viability of different prostate cancer cells, DU145, LINCAP, and PC3. And DU145 and PC3 are androgen independent. And we found like nimbolide can suppress the viability at a very low IC50 at 72 hours. It can induce apoptosis. So we have done the standard techniques like Western blot, looking at cleaved-PAR caspase-3, and we can see a significant apoptosis by nimbolide. Then we looked at the cellular invasion, looking at the matrigel assay. We can see a significant separation of invasion by nimbolide alone, and even in the presence of the chemokine CXCL12. We observed similar effect while looking at the migratory potential of prostate cancer cells using wound healing assay. And again, CXCL12-induced migration was suppressed by nimbolide. Then we proceeded to carry out in vivo studies on anti-cancer effects of nimbolide. So we have developed a transgenic mouse model, which is a transgenic adenocarcinoma of mouse prostate model. This is a transgenic animal model in which we have used this oncogene SV40T antigen, which is expressed specifically in the dorsolateral prostate. So we have developed this model in my lab, and we have used this model because the stages of this model closely resemble the histologically and biochemical nature of the disease in human. So you will see when we use this model, we will see the formation of epithelial hyperplasia at 8 weeks, low pin to high pin grade in 12 weeks, fecal adenocarcinoma 12 to 18 weeks, and differentiated carcinoma in 24 weeks, and followed by metastasis at 28 weeks. And if you castrate this mice at 12 weeks, it does not affect the primary tumor development or metastasis in majority of TRAMP mice. Thereby, it is parallel to the emergence of androgen independent state that we see in men. So using this mouse model, we divided the mice in 3 different groups at 4 weeks, between 4 to 12 weeks, 12 to 18 weeks, and 24 to 30 weeks. We looked at neoplasia, cancer progression, and metastasis. So we also did a pharmacokinetic study and selected this dose, which was bioavailable in the serum by oral gavage to be administered to mice. And control mice were given the normal diet for the period of the study. So if we look at the H&E staining, we found like nimbolight treatment can suppress the formation of thin, well-differentiated carcinoma and poorly differentiated carcinoma. And if we look at the tumor growth and metastasis, we can also see that it can suppress the tumor volume and metastasis to the lungs and livers. So we also looked at the tumor tissues and evaluated the effect on proliferation and apoptosis markers. So we can see an increased apoptosis as evident by cleavage of caspase-3 and also reduction in the level of Ki-67. So we also conducted an acute toxicity of nimbolight. So we saw that there is no significant effect at this dose on the relative body weight, levels of different enzymes like ALT, AST, burn, and also the morphology of the normal prostate is not affected. So then we proceeded to look into the molecular mechanisms. So we are interested in this family of transcription factors, which is known as STAT, which are present in an inactive form in the cytoplasm. And there are 7 different genes which play a very critical role in cytokine-mediated signaling. So we have looked at the role of this transcription factor in different diseases and published a number of articles. So this is our recent review article showing that it can be activated by different stimulus, including cytokines, growth factors, different kinds of TGF-beta, oncostatin M, and binding of, for example, IL-6 to IL-6 receptor can lead to activation of JAKs, which can then phosphorylate STAT3 and induce their dimerization and nuclear translocation. So in case of prostate cancer, we have also found like STAT3 is constitutively active in prostate cancer tissues, which have been derived from the patients. And we can see an increased level of both phosphostat3 and STAT3. And and the role of STAT3 in prostate cancer has been reported previously, showing that it can promote proliferation and acting as a pro-survival factor. For example, it has been shown that if you activate STAT3, it can protect the cells from apoptosis due to androgen deprivation upon administration of androgen deprivation therapy. And it can also enhance androgen receptor function in an androgen independent manner. So looking at the connection between the activation of STAT3 and prostate cancer, we checked the effect of nimbolight. So we saw by Western blot that it can suppress STAT3 activation as well as by DNA binding. We established that STAT3 at tyrosine 705 is the specific target of nimbolight, whereas other STAT isoforms are not affected, as well as phosphorylation at serine 727. We also showed that activation of upstream kinases, phospho-JAK1, JAK2 is inhibited in both DU145 and IL-6 induced LIN-cap cell line. We also looked at the levels of STAT3 regulated gene products by Western blot and real-time PCR analysis. So we established that nimbolight can inhibit STAT3, but there was no mechanism in it, like how it is inhibiting STAT3, it is not known. So we went back to the literature review and found like nimbolight is a very potent antioxidant. So it is more potent than vitamin C. So we wanted to check whether this ability of nimbolight to modulate ROS can be linked with its potential to inhibit STAT3. So we all know ROS plays a very important role in a number of physiological and pathological processes. So if you can say, so ROS level once deregulated due to overproduction of reactive oxygen species or imbalance in the antioxidant system, it can lead to a number of pathological and chronic diseases. So we wanted to establish the possible connection between nimbolight, ROS, and STAT3. So to our surprise, we found Like if we treat the cells with broad spectrum antioxidants like NAC or glutathione, the STAT3 inhibitory effect of nimbolight can be reversed. So contrary to the previous reports that nimbolight is acting as an antioxidant, we evaluated whether nimbolight can induce ROS production and whether this ROS is contributing to the STAT3 inhibitory effects of nimbolight. So indeed, we found that nimbolight can induce ROS accumulation in prostate cancer cells. So if you see here, we will see a significant increase in ROS. And we also identified the ROS species. The ROS species here was hydrogen peroxide and not what we see generally hydroxyl radicals. So we also showed that ROS accumulation is driving the apoptosis by nimbolight. For example, if we scavenge the ROS using NAC or GSH, we can see a decrease in the apoptotic potential of nimbolight. So you will see by flow cytometry and Western blot analysis. So next, we investigated that how nimbolight can increase ROS levels. So if you can see, we have tested these two broad spectrum antioxidants. And if we look at the cellular mechanism, so this glutathione system is the major antioxidant, which is responsible for removing H2O2. So it can convert H2O2 into water and oxygen. And this reaction is catalyzed by glutathione reductase, which can reduce GSHG to GSH. And GSH acts as a negative regulator of redox stress and can replenish the normal functions of the cell. So we wanted to check whether nimbolight can affect the glutathione system, which in turn is leading to increased production of ROS. So indeed, we found like nimbolight can inhibit the glutathione, this expression of this enzyme glutathione reductase, and promote cause an imbalance in the ratio of GSHG and GSH. So it reduces the level of GSH, whereas it increases the levels of GSHG, which is an oxidized form of GSH, and thus leading to imbalance in oxidative stress. So next, we wanted to test that how nimbolight is affecting glutathione reductase. So we did a protein expression by Western blot. We see that the protein expression of glutathione reductase is not affected. This activity of the enzyme in the cell-free assay was found to be affected.
You have got 5 more minutes.
Yeah, I will try to finish in 5 minutes. So the kinetic activity of nimbolight, of the glutathione reductase, was affected by nimbolight in cell-free assay. So in this study, we concluded that nimbolight is directly inhibiting the activity of glutathione reductase, an enzyme which is involved in scavenging of ROS or H2O2 by activating the glutathione system. So as a result, it leads to GSHG-GSH imbalance, leading to increased production of ROS, which can then suppress STAT3 activation, leading to apoptosis, inhibition of proliferation, and metastasis, as I have shown in my previously in vivo data. So recently, we have completed another study looking at the effect of nimbolight in combination with chemotherapy in prostate cancer. What we found, like the patients who are resistant to this androgen deprivation therapy, they are generally treated with docetaxel. So we used this recent study to see whether nimbolight can enhance the effect of docetaxel in prostate cancer cells. So we have used these docetaxel resistant cell lines and treated them with combination of docetaxel and nimbolight, and we can see an increased apoptosis. We also found like docetaxel can induce this NF-kappa B activity in docetaxel resistant cell line by increasing P65 phosphorylation. And if we treat with nimbolight, this docetaxel induced NF-kappa B activation can be reversed. So in this study, we have used 2 different mouse models. So we have developed an orthotopic mouse model with luciferase tagged link-cap cell line. So using this model, we have demonstrated that if we use a combination of nimbolide plus docetaxel, we can see a significant inhibition of tumor growth as well as metastasis to the lung and liver. And we have also done some study, Western blot and immunohistochemistry, and showed an increase of pre-caspase-3 and decrease in VEGF and survivin. In addition to this link cap, which is androgen-dependent tumor, we have also developed another mouse model, which is androgen-independent, using injecting PC3 luciferase cell in the mice. And again, we have tested with combination of nimbolide and docetaxel. So we can see a significant decrease in the tumor burden without any adverse effects. So in future, we are currently looking at some of the other studies whether nimbolide can potentiate the anti-cancer effects of other FDA-approved drugs like androgen antagonist. And we are also doing a large-scale study looking at the pharmacokinetic parameters and chronic toxicity with nimbolide before moving to phase 1 clinical trial in prostate cancer patients. So these are some of our works that we have published, 2 studies on nimbolide, one on oxidative stress in STAT3 model and one in enhancing the effect of docetaxel via modulating NF-kappa B pathway. So in conclusion, I will like to summarize that we are using these pro-inflammatory transcription factors, and we feel that they can be used as ideal targets for cancer treatment and prevention. And I would like to acknowledge some of my lab people who have done this work and my current collaborators. Thank you, and I will be glad to take any questions.
Thank you, Dr. Sethi. So if anybody has any pressing question, then anybody has any question?
Hello, Dr. Sethi, it was a nice talk. I saw the data that you have this nimboloid synergistic with the docetaxel, but can you comment on its synergistic impact on other chemos also, like cisplatin and etoposide, if there is literature existing on that or in your experience?
There are some studies, but I'm not sure whether it is in prostate cancer, but in other cancers, there have been studies that it can enhance the effect of chemotherapeutic agents. So some studies have been published, but our current aim is more to look whether it can also enhance the effect of some of the targeted therapies like androgen blockers, which are being used for prostate cancer in castrate-resistant patients. So that is our current aim. But yes, there are some studies, maybe not in prostate, but other cancers with different chemotherapeutic agents.
Okay, thank you.
Thank you.
So if there is no other question, then thank you, Dr. Sethi, for an excellent talk.
Thank you.
I hope it gives this synergistic effect. So thank you very much.
Thank you.
Second connection. So the next speaker is not available online at the moment. We'll try later also. In the meantime, I will request Dr. Bernard Michael Loeffler to come on this stage and deliver the talk. Dr. Loeffler is from IMM Institute for Mitochondrial Medicine in Germany, and he is going to talk on the use of curcumin, resveratrol, and other natural compounds in tumor therapy, a case report. Dr. Loeffler, please.
Thank you. This is forth and this is back, huh? Yeah. Okay. Mr. Chairman, dear colleagues, I'm very happy to be here, and I would like to thank the organizing committee, especially Professor Bharti, for inviting me. Now it's better. And And I would like also to give special thanks for all the people who have made this conference possible with working in the background, and they did really a very great job. So I would like to start saying something shortly about me. When I was active as a scientist, I had a very broad spectrum of areas of interest where I was working in. So maybe some things, I was very active in endothelin. So in my lab, bosentan, or the trade name is Traclair, was found and developed. And so we solved the first DPP4 crystal structure very fast in our working group. But since 2005, I have changed sides, if you want. I am treating patients, so I am looking at the things from the opposite point of view. And what has been discussed here today, and also on the other INCD conferences I've attended, I try to put in practice for my patients. So I don't know whether I can really give you some inspiration for your work, because what you are doing here, when we put it into practice, is very important. So there is one book about cancer which I love most, and that is the book from Mukherjee. Sorry, I don't know whether I pronounce the name right. And I read to all of my cancer patients this short fraction of his book. It is more in the introduction. And it is telling that cancer is not just a monoclonal disease. It is an evolving monoclonal disease, and every new generation of cancer cells will lead to some cells which are genetically different. And because of this, the standard working and treatment principles are not going to work. And patients have to know that, because the paradigm, one illness, one target, one drug, is a pharmaceutical paradigm, and it is just there to earn money, but it is not there to win, and it is not there to help patients. Yeah, there is not only the cure of chemotherapy, and there is not only the way of the oncology conferences, which are always, and I know that since 17 years, deciding the same. They are not deciding individual therapies for individual patients. They are deciding something which guarantees pharma industry to calculate today what they will earn in 3 years. And that is our problem. That is our biggest problem. And that is the problem why what is discussed here and developed here is not taken into praxis. I will show you here a very simple thing, which I will come back to that, with measurement of circulating tumor cells. You see in the upper right, I don't know where, here. I don't know whether you can see this. You can see here in the upper right, the suggested chemotherapy for a colon cancer patient, which is metastatic. And these are the two chemos which are normally given, and they have an effect of 13% in average, or 21%. So it's clear that they will fail, but that is what the patient is offered. So when we combine this chemotherapy with agents which we were talking about here in the last days, curcumin or so, then you can see that you can improve the activity of this chemotherapy dramatically. But when you look, then you can also see, when you compare just these two lines here, that without chemotherapy, the effect of alternative agents is much, much better as with chemotherapy. So we had yesterday a talk which were going in this direction, that chemotherapy can do something, but also not doing same. And when you combine alternative things, then you can also see that you improve when you are taking more than one drug. And that is all speaking for a multi-drug approach. So then it is always said by standard therapy and standard medicine that curcumin is not working because curcumin has a bad bioavailability. Dr. Sriraj Gopi and me, we have been the only ones comparing so far in a human pharmacokinetic study, 3 compounds which are on the market, and the only intravenous curcumin which is on the market, which is shown here briefly. And as I show you here, so the intravenous curcumin, that is the upper one, and curamine active are nearly the same active, and they are active for 48 hours. That is what we have measured. So you can really use them as a treatment. And for the IV curcumin, there is in the meantime a clinical study available showing its activity. So when it is always, and as that is always what I hear from standard oncologists, we said that curcumin, for example, it's just an example, will not work because of bad bioavailability. This is simply not true. I have contributed to one English book where also Professor Agarwal is in the editors, where I have a chapter about curcumin and how to look at what you can achieve with different curcumin formulations. The other one is in German because German physicians just read German and not English. And to educate them, you have to follow them and to write in German. So I have showed you at the beginning results from circulating tumor cells. I will very briefly go to the two labs with which we are working, measuring circulating tumor cells. So this is the one, and what they are doing is sketched here. So you have a blood sample, and within the natural environment of the blood, you are looking for EpCAM positive cells, and then you are counting these EpCAM positive cells, and then you can look with which agents you can interfere with these EpCAM positive cells and kill them. So the other one technique is a little bit different. They are purifying out of the natural blood with a magnetobead technique the circulating tumor cells. And then they are also measuring it, but they are not measuring the death of the tumor cells. They are measuring the decrease in ATP use. So the cells which are not living anymore, they don't need any ATP anymore. And then it is transformed into a concentration dependent inhibition plot of what you get. So that these tests to measure circulating tumor cells are predictive for the evaluation what compounds can be used in the treatment of a tumor individually for the patient. This has been shown. This has been shown on more than 5,000 patients, but it is not taken into consideration in standard tumor therapy. So as we are using two different laboratories or two different techniques, and this is due to because of redundancy, I show you here a comparison, and I show you that both labs coming to the principally same results. So that means this is a cross-reference that what we are measuring here is really of some importance. So now I come to the case I want to show or want to discuss with you shortly. This is a case of a patient who came to my practice first time 2015, just with some general questions concerning his general health situation and preventive checkup and so on. This patient, the most severe problem of the patient was an allergic bronchitis, which he had since childhood and which was treated with corticoids since childhood. And this maybe is also the reason for why he developed cancer. So he came back in 2019, also just with a different question. He had found his wife, they wanted to get a child and they couldn't. So that was the question, but we made for safety reasons an X-ray of his thorax, and we found that the whole left lung wing was full of cancer. So world changed within 12 hours for this couple and for this patient. And so I was not bound to the general ways of treating patients as this is true in Germany, but also in America. So because I have my private practice, I'm just bound to what the patient wants. And this is a big difference and it is a big advantage. So immediately we started to big biochemical evaluation, and we also started to look in possibilities to deal with this cancer, which was at that time even not characterized. So what I show you here is very general. It is not just for cancer patients. You see here a huge chronic silent inflammation. So all these inflammation markers here are severely up. Yeah, you see, and you see here, this is all these markers, all these different markers, which we also have talked about yesterday. They are all up. Patient has no idea about it, but they are making cancer. So, and then when you look in different parameters, then you see here that this patient has a severe vitamin D deficiency, yeah, makes cancer. So the most important thing from vitamin D is normally not discussed. Yeah, it is free 25-dehydroxy. That is the thing what is finally making the goal. And it is very low. The reference values have changed in the meantime. They are much higher. When we started to measure this, we were one of the first doing that, and then you see down here that 3 vitamins, B1, B2, and B6 are severely deficient. So B1 deficiency makes cancer, makes dementia, makes a lot of things, but it certainly does make the mitochondria unfunctional. Vitamin B6 is involved with the glucose metabolism. If you have none, you have pathologic glucose metabolism. Vitamin B2 is needed for the pyruvate dehydrogenase. So without vitamin B2, no energy production. So it's a disaster. And this disaster you find nearly in 70% of well-nutrition patients which are coming to my praxis. So they are already at the upper level of their income. And even so, they have it with the nutrition which they are having every day. So this is producing not just cancer, it is producing chronic diseases all over. So we made with this patient before, as I said, we did know the final histological diagnosis, already a test for what could interfere with his cancer on circulating tumor cells. That is what you see here. And you can see here already that, for example, curcumin had an effect of 85%. Thereby, it was better as all the pharmaceuticals which are normally used in whatever colon cancer. But you also see that we looked for circulating tumor stem cells, which you can also do with one of these assay setups. And we didn't find at that time circulating tumor stem cells, which was good. That is something I will not go into detail with. So within 5 days, the patient went into surgery. It was a complete removal of the left lung wing, and together with 12 lymph nodes, and it is called an R0 surgery because from the point of the surgeon, he has done everything. No cancer has been. After the surgery, and I show you that just here because you see how much the number of Epcam cells, these are tumor cells, if you want, went up from 150 to 1,000. This has something to do with handling the tumor. Handling it, I mean, when you want to make the surgery, you have to handle it. Then the tumor sends around in the circulation huge amounts of tumor cells. So it could be a signal for metastasis already doing this. Yeah, so one, for example, could think about doing something prevented before surgery or so, but in standard procedures, all these things are not done. So, and I'm showing you here, you know, this case has in the meantime 5 folders. So I cannot present to you here 5 folders, but I present you here results of circulating tumor cells over time, and you can see they are reproducible. Yeah. So even when the number of cells is going down after the surgery back to 350 cells and then to 200 cells. So what you find in terms of sensitivity of these cells is more or less the same. And you see also more or less that chemotherapy is not the first choice. So also shortly after we got the histology, and this patient has a very rare lung cancer. It's about 2% of normal lung cancers. He has a carcinoid, and it is put into the folder of neuroendocrine tumors, even so that specific PET scans, for example, have been always negative on this patient. So, and what we did already from the biopsy, which we got the day after the diagnosis, is we made a severe molecular screening. So we sent the part of this material to a specialized lab, and so the report is 58 pages long, also not something for here, but the summary is standard chemotherapy is not the first option. So that was already clear from this report. Then we had a big discussion with standard medicine whether this result is valid, because it was just from a small biopsy of this huge tumor, and the reality might be completely different. So therefore, we performed from nine of the tissue samples we had from 3, and from the 12 lymph nodes from 2 by random selection. Again, screening for molecular characteristics of this tumor, and all the specimens showed the same as from the first from the biopsy. So, and all showed again that chemotherapy radiation is not the way to go. Even so, the Charité, that is the big Charité in Berlin, yeah. Oh, sorry, I did not translate it. Sorry. I gave a talk recently. They suggested chemotherapy radiation. So, because, you know, in standard thinking, there is nothing else than chemotherapy radiation. So there is no guideline for this tumor. There is no first line chemotherapy for this tumor. And when you look in literature, the literature tells you that you should not do chemotherapy, but the standard is the standard, and it is suggested. And in this special case, yeah, it is the worst what you can do, because the patient before had already lung problems, yeah, with his chronic inflammation, and the risk of chemotherapy radiation for lung patients is that they are developing lung fibrosis, and so they are dying from the therapy. And there are a lot of papers showing that. But you know, when you are just able to think standard, yeah, then this doesn't matter, because normally a lung cancer patient anyhow doesn't live longer than 12 months. So, yeah, and that is, it is unbelievable, yeah, that people who are dealing with this day by day are not able to think out of the box and to realize that there are other options. So we made a lot of additional analyses with circulating tumor cells. This is relatively bad to see, but you can see that we also tested combinations. And at the lower here, you see methadone. Yeah, methadone you might know as therapy for pain therapy. Yeah, and there it is known that when the opioid receptor is mutated, then the opioid receptor has a big impact in tumor growth. And one can see here, methadone is working. So we came to a therapy scheme, yeah, where we heavily increased his vitamin D level. He got resveratrol, he got curcumin, he got once a week a curcumin infusion, and he got methadone, and he got silymarin, because silymarin improves the pharmacokinetic of methadone. So with this, we kept this patient in a good way till November 2020. Control MRTs have been negative, yeah, and then the patient didn't want to take methadone anymore for what reason ever. He has decided he needs a break. And I could not convince him not to do it, so he stopped it. And in February 2021, in the next control MRT, he had a small positive lymph node. It was really small, just 4 millimeters. Normally one would not look at it, but in this case we did. So the patient underwent selective proton therapy for this lymph node, but proton therapy has a risk of, because it's like as if you put an atom bomb in the middle of a city, so everything is exploding, so it has a risk of spreading and it has a risk of creating metastasis. It can be very helpful, it can be also very risky, and exactly for him this happened. He got cutaneous metastasis, and so then the Charité again opted for chemotherapy. At the meantime, the patient had the stamp palliative, that means everything is lost, you cannot do anything anymore. Besides the fact that he's playing tennis, he's doing very well, so he is not behaving as a palliative patient. So, but I mean, that is what they did. And I show you here again results for the suggestions. You see here for the first time the two sites. So this is the next oncology group, and you see here the amplification of tumor genes, which are done with these purified circulating tumor cells. And here you see the results of the suggestions of the Charité in terms of chemotherapy, with combinations and alone, and you see it does not work. It does not work simply, yeah? So, but you know, if you are a Charité professor, then you are in a position with your white coat that you can just disregard everything. Yeah, you does not have to look at real facts. So
five more minutes.
Yeah, I'm ready in a minute. So we have we have remeasured also what we are doing, but we also measured two new possibilities, Incd2022 and Incd2022-Hall. And you can see with these two, yeah, here, and also we have remeasured methadone, and you can see methadone is still in the top, going up to an effect of 70%. So, but these are really promising. These two, they are relatively new, and maybe you know that or not. So we also, again, in one panel measured the suggestions of the Charité with our other compounds, so that one cannot say, so this is one assay, this is another assay, so it's not comparable. And so at the end of the day, we are decided for a combination therapy with Incd2022, curcumin, resveratrol, artesunate, and methadone. So that is a multi-target therapy. And now you might ask, what is Incd2022? And then we are here at a curcumin conference. So this is the target of curcumin. Yeah, this is the death receptor, or the trial way. So this is known since a while. And Incd2022 is exactly something, it's a new generation of therapeutics which is interacting with this receptor. There are at the moment, I think, 126 publications around that. And there is one clinical study, that is this one, showing in human that it is working. So, and we are doing this because I don't have to wait until this has been officially recognized by German standard medicine. That is, as I say, an advantage. So I recommend to you this book from Daniel Kahneman. It is the newest one. It is from 2021, and it has something to do with human judgment. And the chapter 5 is dealing with medicine. And if you read it, you will find out why we have so many problems at the moment with our standard medicine. Yeah, the judgments simply are wrong. So, and so I want to sum up with saying, so we are always in cancer facing individual situations. Every single cancer patient has a different cancer. The classification in receptor positive, receptor negative, HER2 positive, it is wrong. It is too rough. It is not getting to the point. Every breast cancer patient has to look at singularly in her special situation. And if you want to win, you have to have a therapy which is pointing to this single situation. So that means the guideline medicine, which we are still performing, is running in the nowhere. It will be never successful. The only one for whom it is successful is Big Pharma, and that is the tragedy. Thank you very much for your attention.
Thank you, Dr. Lester. So, any questions? Anybody have any question? So individual treatment for individual will be okay, but the number of cases are so much. Do you think it will be possible?
You know, this patient we have treated completely without standard therapy. I have a lot of patients where we are doing a combination. So I have principally nothing against the chemotherapy, but the chemotherapy should not be performed blind. Yeah, the chemotherapy should not be performed because of a guideline. The chemotherapy should be performed on the basis of measurements. So when you show that you have a 70–80% reduction in tumor cells with whatever chemotherapeutic, then yes, then we use it. And then we look for how we can make it more effective by adding, for example, curcumin or resveratrol or whatever else. Yeah. So, but performing a chemotherapy on guidelines, that is the point what I want to make is wrong. You have to measure it in the forefront, and we can measure it.
Okay. All right. Thank you very much. Thank you.
You're welcome.
Can you try the US thing connection? Is it there? What? Okay. Okay, so we are still unable to connect to the person from US. So therefore, there are two industry talk, and maybe we'll move on to the first talk by Subhadeep Mukherjee. He is from BD Life Sciences, Biosciences, and he is going to talk on BD technology and innovation, facts discovered. Self-alternate, please. Please keep the time.
You can come. Good afternoon, everyone. Can you hear me well? Okay. Just allow me some time to connect the system. Okay. Is it still coming? It's in duplicated. I'll just put in slides. Thanks. Again, a good afternoon and a warm welcome to this session from BD Biosciences. I would like to thank first Professor Alok and the organizing committee to give us an opportunity to talk here. It's an absolute honor. So this talk is about the BD technology and innovation. So when I was a customer of BD, I was on the other side during my PhD days. I was always looking at BD, that what they are bringing new in the flow cytometry space, because they are the leader, right? And now I have the privilege to present one of that, this Facts Discover. It's not only an instrument what I'm going to present, but also groundbreaking technology that we'll see now. So let us start with what it is and what innovation it is bringing in. So right now, if we review that the two common ways or broad ways of looking at a single cell. So one, we find a microscopy, right? That gives us a spatial or morphological information of a cell. But if we want to characterize deep, we want to go deep down into the cell functionality, the identification of that, then you might need flow cytometry. So you can go higher into the dimension in terms of how many receptors a cells is that cell is expressing. If that cell is a very rare type, lying deep into or masked into the other cell types which are broadly expressed or broadly represented, then you need flow cytometry to find that. But these two technologies right now are complementary, right? And we do have utilization or benefits of both of them. So of course, the research is going on in this space that whether we can leverage a technology, we can come up with a technology which leverages the benefit of both the microscopy and the flow cytometry. So we can understand the spatial information as well as the immunophenotyping, what you call in terms of immunology or characterizing the cell. So this paper, which was came into the science cover in early this year in February, that talks about imaging, using the imaging information for cell sorting. So this is something new. This is something innovative that came up. It is a joint publication from EMBL and BD. So imaging flow cytometry is not new to market. There are imaging analyzers which actually, utilizes the flow technology and also gives the imaging information. But you cannot take a decision for sorting the cell post analysis so far, unless this publication came in, which talks about the real-time imaging. So you can take a decision for cell sorting. So you can do your downstream experimentation. That can be a sequencing or any culture further. If you're making a hybridoma, you are going for any other drug techniques, drug development techniques, whatever you want to do post identification and sorting of the cell, you can take a decision for sort. So this is new. This is the innovative thing that came in. Of taking a sort decision based on imaging. BD calls it cell view imaging technology. So you can combine this, or enjoy the benefit of imaging along with flow cytometry. So what you are getting here, the high speed, high parameter flow cytometry intact, you are getting the high content imaging data during analysis, and finally, you can use that imaging data along with or separately the way you want it with the flow cytometry data to take a sort decision. How does interface looks like? Let me just do. Okay. So this on the left side of this like slide what you are seeing in the light whole blurred. So this is a very common flow cytometry basic slide or kind of a data what you see in the dot plot, right? You are seeing three different populations of lymphocyte, monocytes, and the neutrophils. So here the extra what we are getting is the image wall on the right side of that of this slide. So that image wall appears simultaneously with this dot plot. So when you are analyzing the cell, when the cell is still moving through the flow cell, it is interrogating through by the laser. You are getting this individual picture of each and every cell the laser is interacting. So you can see, you can visualize, you can see the spatial information or morphological information of the cell along with this flow data, what you regularly get out of a flow cytometer. So what advantages it broadly brings up? Primarily, it is expanding the scope of flow cytometry technology utilization beyond immunology, because flow cytometry is something, a term usually related to immunology. Immunologists are using it the most. But now with this morphological information coming in through imaging, now it can be utilized in the cell biology as well. If you are working in, say, different translocation assays, intracellular signaling like nuclear translocation assays, you will be able to see it, you will be able to visualize it along with the regular flow data, what you do. If you are looking at any subcellular organelles that you can visualize, you can take a picture of that. And also different internalization assays, cell-cell interaction, immune synapse assays. I have a data for that I will show you. Those things which were so far we are not able to measure through flow cytometry, or maybe the measurement was indirect. Now you are able to see it or visualize it directly. And the last one is, of course, on the DNA and cell division analysis. DNA content analysis, ploidy, is something researchers are regularly doing with flow cytometry. But so far, with a conventional flow technique, it is not possible to look into the different stages of cell division, right? But here, as you can visualize it, you can see the different phases of cell division along with the deployed information. So that's what we are adding in with this. How the technology works. So basically, this technology is adapted from telecom industry, telecommunication, broadband. We call it orthogonal frequency domain multiplexing. So it is a very same technology, the way a broadband router is connecting to 5 different systems. So laser is also modulated at different frequencies. So it's not a single laser beam which is hitting the cell. There is a modulator associated with the system, which is modulating the laser at different frequencies. And when the cell is passing through the laser at different, as you can see, this F0, F1, F2, all are hitting the cell in an overlapping manner. And we are getting this waveforms, what we call it, which is generating the image along with the flow data. And how we are collecting this image data, which is generated simultaneously, we are collecting it in a form of a TIFF, T format, and all the TIFF together, because maybe one cell are getting 1,000 like same pictures or waveforms. So all are combined together in a GStack file. So when you are getting the output, you are getting this file .fcs is a regular flow cytometry data raw file what you get out of a flow cytometer. Along with that, you are now getting the GStack, which gives you the imaging data. So if you want to go further down, you can utilize the Flojo software to analyze this imaging data as well. Now let us see that what kind of data we get out of this and what benefits it gives you. So one is on the mouse over imaging for scatter detection, right? So we in flow cytometry, when we are running any sample, we are taking a decision for sort. First, we discriminate that these are our singlets and these are our doublets, means whether two cells are fused together or not. So this doublet information, so far, we are taking that call based on the forward scatter and side scatter data out of a dot plot. But now, as you can see the cell, you can, like the way you are seeing on this right side of the plot, you can see whether you are looking at two cells fused together or you are looking at a single cell. So easily you can take a decision that whether you are looking at a singlet or a doublet. You don't need to do any indirect measurement or decision-making criteria based on the dot plot. On the bottom of the plot, what you can see is a measurement, what you call the index sorting. If you like to sort one single cell in 96 OL or 384 OL plate, it is possible with any regular flow cytometer sorter nowadays. But what it is adding up, that if you are sorting say 96 cells on 96 well, okay, of a plate, then you can see each of this cell which are deposited in the well, and you will be able to understand whether you have sorted the right cell. So you are more confident if you are taking further those cells for culture or sequencing, that you have the right kind of cell in that well deposited. And so that's the addition, that's the quality metrics it is adding on. Tumor interrogation now become more easy, as now you don't have to look into the TILs based on their markers. Here is a mixture of, mix of the TILs and the cancer cell, and based on that image wall, you can clearly see that the spatial information, the morphology of cancer cells are very much different. That is, of course, that is obvious from the lymphocytes, which are more circular in nature. So you can, without any labeling, just with a calcine dye, which is a live dead dye, analysis dye, so you can take a decision that whether you want to, you want to sort TILs or the cancer cells. Cell-cell interaction studies, mostly the immunological synapses. This is really a benefit which we are adding on with this imaging data, because so far this dot plot is something what we are getting out of a conventional flow cytometer. And here on the x-axis of this dot plot, on the right left side of the plot I'm talking, we are seeing the CD3 positive T cells and the CD14 positive monocytes. So if you look into the image down there, so LFA1 is expressed, this green cells, which is expressed by the T cells, and ICAM is generally expressed by the monocytes, right? So now this data from the dot plot, now if you look into the imaging, you will be able to understand that whether an immunological synapse formation is happening or not. because on the 3 images, if you look into the first image, you can see there is a yellow light coming in, right, from the middle. So that is the evidence that there is a LFA1 and ICAM interaction has happened, right? And that is giving us the yellow light compared to the passive synapse where we don't see any yellow light coming in. So as this is a direct visualization, direct measurement, it gives you more confidence or opportunity to study the immunological synapses. Same goes for the tumor cell killing assays, which are on the right side. You can see the usually the like if you're working on a TILs or something, some other cell cell therapy, if you're doing, you'll be able to see the interaction of the lymphocytes with the cancer cells together. So no indirect measurement, directly you can see the data. Cell-cell interaction studies, as was say translocation studies, what I was telling, any functionality, any kind of pathway study, if you want to see, you can do in this way. So nuclear translocation assay, NF-kappa B translocation to nucleus. Conventionally, we do it with indirect measurement. Now, here on the image wall, as you are seeing, that when in the control or unstimulated, the green one, okay, which is on the left side of the image wall, compared to the right side of the image wall, which is the TNF treated stimulated. So all the right side of the image wall, what you are seeing is the yellow light coming in, and on the left side, which are unstimulated, you can clearly see the nucleus, which is red, and the outside the NF-kappa B, which are FITC marked, which is green. So red and green can be clearly distinguished because NF-kappa B is on the cytosol, but with TNF stimulation, what you are seeing in the right of the image wall, all are inside the nucleus. So they are together. So we see the yellow light coming in. So this is a direct measurement, or for any nuclear translocation or any pathway analysis, if you want to do, you can do in this way. So of course, so far the data I'm showing, and this is I'm telling you the advantage of this technology of cell view. Of course, there is an instrument behind it. So this instrument we call FACS-Discover S8 cell sorter. How it looks like. This is the instrument. It's a benchtop instrument, as you can see. All the laser modules, fluid modules are below the table, and the workstation is there on the top of the table. As I was telling, that what it does, along with the keeping the flow cytometry data integrity as it is adding the imaging information to take the sort decision. In the sorting part, it's a 6-way sorter. So it retains all the other benefits of any BD sorter, like automated stream setup or automated sorting criteria, but along with it's a 6-way sorter. So you can sort 6 different populations together based on the imaging criteria, based on the flow cytometry criteria data output, or combined measurement. So this is the correlation, what I say, the benefit further after sorting. So you can take with the cell morphology or the protein expression or CD marker analysis data. If you want to do downstream multiomics analysis with BD Rhapsody system, you can do that after sequencing in, say, single cell mode, or maybe in a bulk sequencing. So if you want to sort a Treg cell, you can do that based on the imaging criteria, and then you go for multiomics to understand how differentiated further the Treg populations are. So that is how it is connecting to the further other technologies for discovery analysis. And in the CD marker part, the new part, which is feature which we are giving you, it's a spectral flow cytometer. So it's not conventional. So there are 78 spectral detectors out of 5 lasers which are this equipment is actually having. So 78 spectral detector plus 6 imaging parameters plus FSC and SSC. So that is the total 80 plus detectors what you are getting out of this instrument. And when we are giving you 78 spectral detector, that's a huge number. That's a real high dimensional flow cytometry we are talking about. So we know that we also need to give you the dyes, because the reagent availability is always a question. That we can go high parameter, we can go into 70, 60, 70, 80 plus detectors, but do we have the reagents? So keeping that in mind, BD launched the spectral series of dyes, the BD Horizon Real Yellow, which are laser specific dyes. So if you are conversant with flow cytometry, and if you're using fluorochromes like PE, phycoerythrin, you might be aware that it is cross laser excited. Like it can be excited by the blue laser as well as the yellow green laser, which creates sometimes a problem in compensation or data analysis. But all these real dyes, whether it is real yellow or real blue, these are laser specific. So when we call it's a real yellow dye, these are all excited by the yellow green laser, which is 561, not excited by any other laser. So cross laser excitation problem, which can further contribute to the spread in data, that is eliminated. And along with the image sorting, you can use this spectral information for sorting as well, as was mentioning. So this is my last slide, that taking all this together, the cell view image technology, the systematic approach to sorting, spectral analysis and sorting, the software innovation in terms of Flojo, because when we are bringing in, as I said, this Flojo Lens plugin is also now inbuilt within Flojo. So if you are using Discover, you are using those, getting those image data, you can use that Flojo software to analyze. And finally, this combined data is advancing, the sorting module, the way so far it is, absent, I would say, in terms of using the real-time imaging for sort decision. So I would like to stop here. If you have any question, I'd be happy to answer.
Thank you, Shobhadeep. He will be available for discussion if anybody has, but if anybody has any very important question, very pressing question, I'm sure he has given a lot of information. You can discuss during the lunch time. Thank you very much for the
Thank you very much
for the information. So now we have another industry talk from Biorad, I guess. Digital PCR. Yeah, so new kind of PCR, you can buy the machine.
Hi, very good afternoon to everybody. Hope I'm audible. Yeah. So, hi, this is Dr. Abhijit. I represent Biorad Laboratories, and I'm here today to give a brief talk about a newer technology in the market called as a droplet digital PCR. So, just in brief, what I would be doing is I'll be discussing about the technology, its applications, and what are the advantages that you can have as compared to the conventional technologies. So I would just request if anybody has any questions, concerns at any point, you can stop me. I'll try my best to address it. So just in brief, I'm sure all of us know this. This is just how the life cycle of a PCR has evolved. All of us know what we talk here of a first generation PCR. We put a thermal cycling reaction, we run it on a gel. If we see a band, the DNA is present. If no, it's absent, right? Then came the second generation of PCRs, which is called as a quantitative PCR, or more commonly called as real-time PCRs, okay? Where it's always assumed that we get to a quantitative value of your nucleic acid. But a very important thing to take a note here is the second generation of a PCR, or the real-time PCR we're talking of, is always a relative quantification. You need to have certain standards, you need to have certain housekeeping genes, controls, only then you have a deduced value coming out of your data, and you come to a value which is telling you a quantity of nucleic acid, okay? So a lot of processes involved, a lot of calculations, a lot of discrepancies. Then we come to the third generation of PCRs here, which we call it as a digital PCR, or more specifically, because I represent a brand, which is called as a droplet digital PCR. Which the major advantage is it gives you an absolute value of your nucleic acids. So the data that you get is in copies of your nucleic acid per microliter of your sample. You don't need to run any standards, no need of housekeeping genes, no need of calculations. It's a fully automated system that will give you a precise quantity of your nucleic acid in the sample. Okay. Moving ahead, the major step of differentiation of how a digital PCR is different than a real-time PCR is the principle of partitioning. Okay. So in this case, what we're doing is we have a same reaction mixture that we make for a real-time PCR. That 20 microliter reaction is now split into 20,000 compartments. Okay. And now there are 20,000 parallel PCRs which are happening, which are working together. And each of those 20,000 droplets are counted individually. So now you have a combination of 20,000 readouts from 120 microliter reaction. Okay. Just as an example, if I would have to show you this, now if you're walking or if you're standing on a pond and somebody asks you a very basic question of tell us how many are yellow fishes, how many are red, how many are orange, it's practically impossible, right? But if we now remove those and put them in individual bowls, and then if you're asked to count, it's easy, right? So this is exactly what we're doing here. We have a compartment, we have individual compartments which are now amplifying your nucleic acid, and then you detect and calculate each one of them individually. Okay. So the major advantages that you get with the ddPCR as compared to any other conventional quantitative PCR technology, first we're talking of a precision. Okay. So we have umpteen publications where we have shown that even a single copy of your nucleic acid, let it be a DNA or a RNA, is precisely quantified, is precisely given as a data point. Okay. Coming to the second point, which I already mentioned, this technology is an absolute quantification method. You don't need standards, you don't need reference genes, no calculations involved, no interpreted data. It is a direct quantity of your nucleic acid which is presented to you. Okay. Third point coming to is the multiplexing. This gives you a flexibility to use a dye-based chemistry also or a probe-based chemistry also. And in either of those cases, you can do multiplexings. Okay. So there is no need to worry that you have to run at one gene one time. You can have multiple genes in the same tube and you get precise quantity of each one of them. Okay. Next advantage, sensitivity. I already mentioned it can go up to a single copy detection very precisely. Again, just to add on to a point here, the advantage here is even if the quality of your nucleic acid is bad, if you have degraded DNA, if you have like liquid biopsy, if you have nucleic acid from your paraffin blocks, right? Even those type of nucleic acids can be precisely quantified using this technology. Okay. And the last and the most important point is tolerance to inhibitors. Even if your sample has tons of inhibitors, it is not going to work on any other technology, but this will still precisely quantify your nucleic acid and give you a precise data. Okay. So these are the major advantages of having this technology. I'll just quickly explain you the workflow, what happens here. So in the first step, what we simply do is we have a normal reaction mixture which you make for a real-time PCR. You have your primers, probes, nucleic acid, supermix, and that is now split. This is exactly how it works. So there is an oil, there is your nucleic acid reaction mix. It makes droplets. It makes droplets of exactly same size, same volume, right? And now these droplets we put back in a PCR plate which goes in a thermal cycling. And now that's where thermal cycling happens. And then we have another small instrument which looks like this, which is called as a droplet reader. So my friend who gave a talk before this from BD, right? So all of us understand how a flow cytometer works, correct? So this reader is exactly like a flow cytometer. Only difference being a flow cytometer counts cells. A same principle here. This counts each droplet, right? So there are multiple lasers sitting inside the reader. Each droplet is going to pass through in a collinear manner. And there is a precise detection that happens. And that's how you get a precise quantity of nucleic acid. And the data that you get is something like this, yeah. So a scatter plot that you get on a real-time piece or a flow cytometer, a similar scatter plot is given here as well. So this, what you see here, this is the data from per well, okay. So each of this dot represents one droplet. So you're not missing onto anything. The droplets which do not have your nucleic acid will have no fluorescence. They would sit at the bottom right here, right? And the ones who have fluorescence are here. So that's how they are scattered. That's how they are plotted. And a similar thing in a 2D, you see it here. So anybody who understands flow cytometer, you have a FAM positive, you have a hex positive, you have double positive and negative. So similar data is there, and the software then interprets further, converts the digital signals into a quantitative data, which is given to you precisely. Okay, I would skip this because of time constraints. Yeah, this is just again a 2 months back old data still. So today we have got close to 6,500 publications using this technology. Okay, across the globe, across different applications. In India, we have close to 100 installations now with this technology. A lot of people getting advantage. So those of you who are working on any kind of a nucleic acid amplification process, I would urge you to take this into consideration. And this is how the life cycle has gone. So Bio-Rad has been a proprietary manufacturer of this technology. It's been 10 years, it's been just a decade now when we had started this. And these are the different product lines we have crossed over a decade. And with a lot of pride, I can say we have expertise, we have support, we have global knowledge base, which can help you understand this better. Okay. And these are just the applications broadly in which this technology has been used, starting from viral titers to agriculture to food biotechnologies to water detection, to somebody working on gene expression, mutation detections, all of it. Basically, any kind of nucleic acid work you want to do, this can answer your concerns. Okay. What I'll now do quickly is a couple of slides where I would just show you a few snapshots from few publications, okay, where this technology is helping. So this was one of the publications which is from Harvard, if I'm not wrong, where a precise 1 copy to 1 lakh copies of your viral were loaded in the sample. And that's how a precise detection of that was done with this technology, right? Again, coming to next study, this is again from the publications where this was carried on across 5 different countries in probably 5 different parts of the globe, right? Similar sample was sent, different machines, different time points, and this is how a precise data was represented at the end of it. This was close to some 5,000 odd samples across different works. Okay. One more very important, I would say, feather in the hat for us, the National Measurement Institute in Australia, which does validations for most of the controls that we get for different instruments, right? So this is an instrument which is used for validations of those known controls, right? So this is one of the data which they had published across some 5 lakh samples. And you see in the second graph, less than 2% CV. So this is how reproducible, this is how precise this technology is. Okay. Again, a very, very challenging application for those of you who work on copy number variations. If you want to differentiate between 1 copies, 2 copies, it's still easy, right? But when you go to differentiating 5 versus 6 copies or vice versa, right? That's when the problem comes. And this is the ddPCR, again, one of the publications where you see the copy number differences. And the last part is when the same was done on a well-optimized real-time PCR technology. You see the variations there and you see the precision with the ddPCR. Again, this was from Stanford. So, I mean, you can be rest assured of what was published is what was done there. Okay. Again, one of the very critical application when someone goes to single point mutations, right? Below 1%, a real-time PCR is never going to detect your mutation frequency. And this is again from one of the publications where right from 0.001% mutations all the way till 1% by multiple users has been verified. And this is the precision with this technology is detected and quantified that nucleic acid. A last point I would focusing upon again, which becomes a concern for a lot of us who are using a real-time PCR is inhibitors, right? Our reactions don't work and we don't know why it is. Again, so again, this is from one of the publications where a lot of inhibitors were purposefully added to the sample just to check how rigid this technology is. And as you see, you have media, you have HEK cell lysates, you have DMSO, EDTA, and still the technology has precisely quantified without any variations and given you the quantity of nucleic acid. And just the last part of it, where I was talking about the multiplexings, there are certain publications where with a dye-based chemistry also, people have done multiplexing up to 4-plex assays in a single tube. And these are a few publications. And this is from a probe-based chemistry, where you're using a TACMAN chemistry or any other FAM hex with TAMRA or any other quenchers. This is how people have published up to 8-plex assays in a single well with this technology, right? So yeah, that's how it is represented for the single positives, double positive, and the negatives. And just as a last thing, if someone is using a real-time PCR, I mean, it's not that you have to do something great. So this is a tech note and a publication that explains you how a well-optimized real-time can directly be replicated for this technology. You don't have to do any efforts. just with the added advantage, like you don't need standards, you don't need references, inhibitors, and the most important part here is your LOD and LOQ is always the same. Your limit of detection and limit of quantification is always the same. So somebody who's use a real-time PCR, your LOD will always be higher because any fluorescence coming out of your machine, it will get captured, but that cannot be quantified. But here, whatever fluorescence is coming in, whatever nucleic acid quantity is there, the machine will precisely detect as well as quantify that. So that becomes a major advantage. And I would like to stop with that. If there's any questions, I would be more than happy to answer.
Thank you, Abhijit. Anybody have any question?
Yes, ma'am, please. I think this question, yeah.
As you showed, the digital PCR has so many advantage over the real-time PCR.
Correct.
But what's about the cost effectiveness?
Ma'am, yeah, very good question, ma'am. I will tell you point blank, this becomes cheaper as compared to even a real-time PCR. In CPT, what we call as cost per test. Because in a real-time PCR, practically to get one meaningful data point, you need to have your 3 wells minimum as your control gene, 3 wells for your sample, your negatives, positives. So practically close to 10 to 12 wells for getting one meaningful data, which is still a relative quantification. Here, your one data point is your quantified data precisely.
I think it will be less cheaper.
It is cheaper as compared to
but
real-time
when
PCR.
we have to analyze so many samples. But if
Yes.
I have very few samples, I have to do the digital PCR. In that case, I think it's not cost effective.
No, ma'am, it is still cheaper. It is still cheaper because, I mean, I did not get time to explain you the entire workflow. You can practically run one sample to 96 samples at the same go at the same time. And this is, ma'am, there is no difference. Your real-time PCR reaction, what you make is still going to remain the same. The only part that is different here is your partitioning in the first step where you're making droplets. That's the only additional point. Other than that, your reaction mix and everything is the same that you do for real-time PCR. So there is practically, I would say, a 10% increase in your price if you're talking of a single well. But when multiple wells are used there as replicates, here it's a single data point from a single well. So all of that is nullified and this becomes a cheaper technology compared to real-time PCR in terms of running cost.
Experiment is cheaper, only machine is costlier.
Yes, rightly said, sir.
Yeah. 60 lakhs.
So there are different models. There are different models depending on what you're looking at. I mean, so it should be somewhere close to 70 lakhs, I would say, 70, 75 lakhs. It's a workflow. We are talking of 4 different instruments with consumables, everything as a package.
So droplet has oil on the surface.
Correct, sir.
But when you heat it, don't they get
No,
bigger
no,
droplet?
no. So this is, I mean, we talk it as an oil, but they, it's a proprietary product. It has a lot of things added to it, which maintain the integrity, right?
Yeah.
So there are, in fact, a lot of publications also where we have shown how the integrity is maintained and how each compartment of a 20,000 droplets is same, is uniform, is of a same volume.
Thank you.
Yeah.
So
yeah.
I have one quick
Please,
question. Sir. Do you have also any kind of provision for reagent rental?
We can talk about it, sir.
Okay.
We can talk about it.
Because we are thinking about in our hospital to
Okay,
put
sir.
up
So
the
I am here. My colleague is here, sir, Miss Akansha, and we have a stall. If we can find some time after the talk, we can have a detailed discussion.
And also, what are the different hospitals where you have put this machine is installed?
I think he is available and people can discuss with
Absolutely.
him in the lobby or during the lunch.
Yes.
So I think with this, the session is over. Thank you, speakers who are here. And thank you, everybody, for joining this session. Thank you very much.
I think. Hello. Hello. I thank all the speakers. And now I request Professor Yogen Singh to facilitate all the speakers. Professor Loeffler, please come. Dr. Subhadeep Mukherjee. Mr. Abhijit Dikshit. And organizers will post the memento to the speaker, Professor Gautam Sethi, who has presented online. Thank you. That's Thank you everyone for their valuable time. Thank you.
अगले सेशन